Hello everyone, my name is Wilfredo Garcia and with Saul Ibarra and Juan Gonzalez we will be going over blood flow research papers. The first paper was on a computational model of blood flow. This paper aimed to determine a computational model that could be used to generate microvascular geometries. This was achieved by creating a mathematical model for microvascular geometries and blood flow. The characteristics of blood flow predicted by the model were similar to those found in experiments on mice. However, there was a scalability concern as humans have red blood cells that are 30 times larger than those of mice. The next article is on viscoelastic transient of confined red blood cells. The paper aimed to study the response time of blood cells confined to microchannels in vitro. The results were obtained by using a theoretical analysis numerical model, and they demonstrated that the effective viscosity of blood is not equal to the membrane viscosity as previously believed. The next article is on parallel modeling of cell suspension flow. The paper aimed to create a hybrid simulation model that could be used to study the behavior of cell suspension blood flow. It used a combination of particle dynamics and immersed boundary models to create a parallel framework. And then it used inflow and outflow boundary conditions, creating a model that was very effective at simulating cell suspension flows through complex structures. The next article was on the startup shape dynamics of red blood cells. This study aimed to provide the first quantitative results of the behavior of blood in transient shear flow. The study uses high-speed video microscopy imaging of red blood cells flowing in silica cylindrical capillaries. The results demonstrate a good agreement between the data and the predictive time found in micropipette experiments, and the model could distinguish between red blood cells with different membrane deformability.
I'm Saul Ibarra, and I will be going over the following 4 articles, starting with lingering dynamics and microvascular blood flow. The study was conducted to better understand the lingering dynamics of red blood cells in microvascular networks. In vivo analysis was employed, where researchers analyzed the microvascular system of living hamsters using varying diameters and bifurcating angles. The data showed lingering at apex of bifurcating angles, deformations of RBCs at bifurcations, void durations were measured, and the results aligned with prior in silico studies. The second article was to study the shear rates, flow patterns, and coagulation factors that lead to platelet activation and aggregation, which may lead to an arterial blood clot. Experimental and numerical methods were used to investigate these patterns. The results show platelet activation directly increased with the time of circulation shear stress and time of exposure to shear. The third article is on the study of biophysical characteristics of RBCs traversing microfluidic channels. In order to quantify the deformation of RBCs, they combined methods of RBC optical tweezers and flow experiments with the corresponding simulation based on dissipative particle dynamics. They observed up to 200 longitudinal axis deformation of red blood cells compared to the average diameter. The fourth article studied the flow-induced platelet activation, which prompts complex philipod formation. A multi-scale numerical model was developed to simulate this activation process where dissipate particle dynamics was employed. They were able to determine the length, thickness, and philipod numbers of the platelets.
Hi, my name is Juan. This article aims to highlight the advancements in blood flow imaging and computing, starting with color Doppler ultrasounds, which are used mainly for imaging of flow patterns, TMRI, which is used to measure velocity vectors of the flow and is limited by the computing power. And finally, CFD, which uses both and is able to use image-based modeling for specific real-world models. This study aims to model effects of shear flow in red blood cells and their dynamic properties using a dextran solution to vary the shear flow and incubating cells to simulate rigidity. This study found that increasing in shear flow increases the orbit angle of red blood cell. Rolling of a red blood cell is due to the finite membrane elasticity. Next, this study modeled the flow and deformation of red blood cells through curved vessels. It used a particle-based method to model the flow in small microvessels. It determined that the velocity profile of the flow is skewed towards the inside of the vessel's curvature. Also determined small curvatures have negligible effects, while large curvatures deform the red blood cells at the peak of the curvature. Finally, this study simulated blood flow by coupling fluid flow and particle interactions by combining the immersed boundary method and discrete element method. It used a reduced order red blood cell model consisting of overlapping constituent spheres with flexible bonds to simulate the deformation of the red blood cells. And it also used single and multiple red blood cell models to show that the shear rate increases the deformation of the red blood cell and that they concentrate towards the center of a vessel regardless of hematocrit, as seen in this illustration.